The next question, who is Leonard Hayflick? If I were answering, I'd say that, well, that was my professor at Stanford University, and I know him very well. In fact, we just had an email exchange recently about the Nobel Prize winners who were his students for the 2009 Physiology and Medicine Nobel Prize, and what is his limit. You know, when Leonard first identified this problem, it was contrary to conventional wisdom, because there were people like Carrel who were working with chickens' cells in media and inadvertently contaminated the culture medium for the nutrition for the cells to grow with new cells. And so it appeared to him that cells of all sorts, not just cancer cells, were immortal. And so the conventional wisdom at the time was immortal cells. And when Hayflick tried to reproduce that experiment, after fibroblasts hit 60 plus or minus 10, they were not able to divide mitotically any further. They came to a dead end. So that is the second question. 60 plus or minus 10 fibroblast mitotic divisions. And then, okay, what's the difference between induced pluripotent stem cells and other types of embryonically derived cells? What would you say about that?
What would I say about that?
Yes.
Well, this is Michael West explained all of it yesterday.
Yes.
So if you're at the meeting, but again, this is probably not going to be in the general domain of the general public. But if they had, I mean, if they just give you an answer like the doctor gives an answer, well, embryonic cells are derived from embryos. Well, obviously they don't know what's going on. Leroy Hood, that's a, sorry. Oh, I'm sorry. The issue of Leroy Hood. I mean, this is actually a very tricky question. This, at least according to Steve, this professor Leroy Hood should have received a Nobel Prize. Without him, we would not have the genome unraveled. And he had the sequencing technology to make it all happen. Now, I mean, this is getting off topic a little bit, but.
Exactly. He was our professor at Caltech when the technology was first developed.
Right. Let's.
Craig Venter.
I'd like to make a comment. I'm going to make an oblique comment here. Why do patients come to see me? Why do they come to see any anti-aging physician? They don't ask me about stem cells. They don't ask me about telomere length. They come to me because they don't feel optimal. They don't feel me. That's the comment that I get all the time. I don't feel like me. They're distressed. This is about functional medicine. This is about restoring you back to optimal functional health and well-being. It's different than conventional medicine. When you go to your physician, all they're interested in, you are sick, you're not sick. If your lab is okay and you're not sick, you're a complainer. You're a worried well. You're out of here. And that doesn't help you. That's not what this is all about. So I think the more important question to me that I want to raise to the rest of the panel is, why do they come to see me or any other anti-aging physician? And what are the qualities that anti-aging medicine has to offer people that is a different approach, that offers different solutions, that's going to help you, the motivated individual, achieve your goals?
Thank you very much, Phil. I think what we want to do is get through this. And what Carlos was proposing is, if the patient didn't know you and he knew 2 or 3 or 4 of the anti-aging physicians, and he or she were trying to pick one, this may be a very good way just to test their knowledge. And then you can go into the commitment, all that stuff. But let's go through here just quickly. We don't even need to read them anymore. Let's just put them up.
I'm going to give some answers quickly. The first human genome actually was Craig Venter's. And the number 2 human genome shortly thereafter, also for nearly $1 million, was Watson of Watson and Crick.
Are these going to be published on your site, Carlos?
It's actually on GRG.
On GRG?
It's on the GRG website.
Give us the URL.
Okay.
The second genome that was done was the one done at by NIH, essentially. It was announced, they were co-announced, and that involved the best of luck.
No, I know what you're saying, but that was a mixture of many different people.
Okay,
What
again,
we're talking about here is
excuse me,
a
Robert.
single person.
I'm sorry, excuse me, Steve and Robert. We're just trying to test the knowledge of the patients. Let's not debate each one of these questions, but let's give everybody an opportunity to tell them what's going to benefit them and how you operate differently from a traditional MD.
Okay, and SNP, single nucleotide polymorphisms, are important in the rate at which somebody may be aging. The epigenome is the methyl groups that are decorating the DNA that determine differential gene expression, how you can transfer genes to alter a person's defective genes. There are various viral vectors and other new techniques that are being discussed. How do animal and other species data show regarding deletion of IGF-1, insulin signal path, and so on. There are a lot of interesting new data items that were discussed earlier yesterday in this meeting that I think an anti-aging physician ought to be aware of. And so we're done almost, well, how can human growth hormone help? And can it burn through? Like somebody said yesterday, pressing the gas pedal to the metal in an old car. In some people, it may be dangerous. Supercentenarians,
Okay,
of course,
excuse me,
is
Steve.
my area.
Let's just put these up here, let people read them, and
Okay.
copy them. And then what's your URL?
The website is www.grg.org/resources. And then you scroll down and you'll find a link to anti-aging doctors.
Right,
In fact,
and-
on that, after these questions, is a list of names and addresses of
Excellent,
people
thank you.
throughout California.
And the patients won't need to know the answers. They just need to get answers from their doctors. Okay, so let's move on to the next.
You want to rush me through, and we have an hour, I thought.
Well,
okay.
yeah, but we have a lot to talk about.
Okay. Which method of life extension and how you would treat obstructive sleep apnea, how you deal with smoking, alcohol, gambling, all of these are very important questions in anti-aging and age management medicine. And I think we're going to talk a lot more about vitamin supplements and libido and various drugs for erectile dysfunction, or when do you give testosterone or DHEA to women? How do you deal with obesity? What kind of fitness program do you have? So let's go now back to the second question that we had before the list of 20 questions. And,
Actually, I'm going to interject here for a minute, Steve, just to pull it back to a little more mainstream. We've just made an executive decision, and then we can get back to the list. Just a quick question. This is actually directed at probably Phil Miller and Joe Mercola as much as anybody. I'm a fitness and health magazine editor. I know what people come to me and why they ask questions to me. I'm just wondering, and Joe, I think you with your huge internet presence and savvy in that regard, if in particular, I don't know that you're known as an anti-aging guy, but for those people who do come to you for that, what would be the top things that they're looking for?
You're right. I'm not known as an anti-aging doctor. I'm known as a wellness physician. And actually, I was just nominated for an innovative name change, or one of the top innovators for wellness off Huffington Post this week.
And you need to put this thing right up there.
Okay, there we go. It's really close. So the people come, I don't see patients anymore. I stopped seeing patients 5 years ago. But my focus is on wellness, to get people healthy. And the strategy is to, as we all know, a lot of these innovations and developments are not ready yet. They're going to be ready sometime down the road. We want to maximize the amount of degeneration that we have so that we can optimally utilize it. So that's what I focus on. And I don't really do any specific interventions. I'm here more of I was excited to attend and glad that Dave invited me because I'm really a student. And I'm one of those, when Bill gave his presentation on telomeres, I was clueless. I didn't even know about telomeres, the truth. And it was a real epiphany for me to have that understanding. My focus is to get people healthy with exercise and optimal food and really minimize the damage and to address the emotional components. There's Bruce. I'm a big fan of Bruce Lipton, and he has one of his main focuses is epigenetics, and to use the emotional components to actually upregulate and and improve the genetic structure with just emotions. It's just profound, absolutely